Good evening, everybody. I want to welcome you, everybody, at 19th of January for this CCAM conference, the extended one. Every Wednesday, almost, we have this conference. And I hope you have I hope you have a nice week. And I have an echo. One moment, please. Sorry. Somewhere a program problem, but I will to give the word to John Ellis. Probably that is better than I keep quiet for a moment.
Okay, thanks, Harman. It sounds like you're having some technical difficulties. So yeah, my name's John Ellis, and I'm a lecturer at the University of Plymouth, and I'm on the scientific board of CICAM. And it's my pleasure tonight to introduce Sonja Leidenberger and Nicholas Norström, both from the University of Skövde in Sweden. And just to give a quick introduction, so Sonja is an ecologist. She does work ranging on a range of different questions, from life history strategies, genetics, species distribution modeling, the climate change, and she's worked on a range of different systems, from aquatic species, invasive species, as well as bees. And in her private life, Sonja started beekeeping several years ago, and it became clear to her that she wanted to keep the dark honeybee, Apis mellifera mellifera, as a pollinator in her garden. And when she started doing this, she met with many different opinions from beekeepers on bee subspecies. So as a scientist, she started looking into the research on the topic, and was surprised to see relatively little research about different honeybee subspecies. And this prompted a research project on dark bees. And she's now working with the Swedish Dark Bee Association, the Nordbyförening, Norden's ARC, and also collaborating with partners in Norway from the Norges Biraktalag. And this is on a 3-year project funded by the EU INTERREG Sweden-Norway program. And joining Sonja tonight is Nicholas Norström, and he's also a senior lecturer at Skövde, and his research focuses on behavioral ecology, theoretical ecology and evolution. And he's also worked on a range of different study systems as well, from host plant selection in insects and fisheries science, and now on honeybees and bee behavior. And I would also just like to say, on behalf of CCAM and all the attendees, we'd like to congratulate Nicholas, because last night his wife gave birth to their second daughter. So it's great that he can still be here with us. I imagine he's a bit tired, but not as tired as his wife. So on that note, I will be quiet now, and I'll hand over to Sonja and Nicholas for our talk, and I'll be back for the question period. So over to you, Sonja.
Thank you so much for this nice introduction. And thank you also to the whole committee for giving me the chance to present our project. Like you correctly said, we are at the University of Skövde, my colleague and me, and it's not only Skövde University. We get the project funded by the European Union, and it's a cross-border project between Sweden and Norway. And Norden's ARC, the Wildlife Park Zoo at the Swedish West Coast, is the project owner at the Swedish side, and Norges Biraktalag in Oslo is the project owner in the Norwegian part. There are also 2 universities joining us. It's Umeå University, far in the north in Sweden, and Gothenburg University at the Swedish West Coast. So why the INTERREG Bee project called in Swedish Bistånd? The main goal was that we want to collect systematic data and documentation of the honeybee in comparative studies, and especially over a long time. Because if you go out and looking for data and looking up research work, it's often maybe only some colonies were studied, or for a very short period. And different honey species are known to have different characteristics, like in pollination effects or finding the food or overwintering strategies or honey production. But most of this knowledge is a kind of anecdotal basis from mouth to mouth or observations by beekeepers. And there is a big need for really scientific data collections, and especially over a long time. The results will improve, hopefully, the future beekeeping. We know that there's quite a lot of going on out there with climate change, and it's important to protect our pollination as a very important ecosystem service we have. Otherwise, the world would look very different outside there. And it's also, hopefully, a part of benefits the local biodiversity that we have. In this 3 years INTERREG project, we collecting quite a lot of data. We studied the phenotypic variation, so observable characteristics and traits of honeybees. And we collect life history characters like age at first fly, mortality of juveniles or adults, flight activities per day, per season, weight change over the year, winter weight loss and winter survival. We was looking how the bee cluster move in the winter over the frames by using thermophotography. We're looking for the swarm behavior. Wind index measurements were done each year. We counted the Varroa mites. We looked at the honey production. My colleague at Gothenburg University was looking at honey and pollen analysis, and the other colleague in Umeå University was doing the nosema counting. And all this data we set in relation to climate data that we were collecting, like light intensity, temperature, rainfall, wind speed, and wind direction. So we collected during 3 years quite a lot of data, and we started, of course, to analyze this and want to present you our first results today. If you're looking in bee research, you mostly find Apis mellifera. So it's not so often that the researcher really defines the subspecies. This map, I think, we have seen different times now during the SICAM conference. But you can see that Apis mellifera has a really broad and huge range, natural range, and is also a kind of link to this climate, while the other subspecies more preferred to the Mediterranean area where it's warmer, and there are no species far up in the north where it's colder. But even in Sweden, we imported 2 other subspecies. In the 19th century, it was ligustica, but also carnica, because ligustica is known as the gentle honeybee species, often for new beginners in beekeeping. Carnica is rapidly adjusting of their brood to nectar availability, especially in spring. And the other one is the hybrid Buckfast that you can find here in Sweden. It's from Brother Adam, who was crossing bees from places all over Europe, Near East and Northern Africa. I will give a short introduction to the black bee situation in Sweden, because after my knowledge, this is the only talk that comes from Sweden. So for those that are not familiar with this situation in Sweden, the green line here in the map is showing you the natural distribution of Apis mellifera before 1750. So far up in the north, there was no natural distribution. Here in Sweden, it's the Nordbyförening, the Swedish Apis mellifera Association. And they tried to rescue the last existing 500 or 500 to 800 colonies at the end of the 1990s. It was Ingvar Arvidsson's work, and then he started this association. Today, you can find around 1,000 to 1,200 colonies. And we have several lineages that could be saved, like Sveg, Skedvik, Bjurholmen, and so on. And the goal is to at least get up to 2,000 colonies out there, because in Sweden we have nearly 85,000 registered honeybee colonies, like the last statistics from the Swedish Board of Agriculture. Most of those colonies are Buckfast in Sweden, so that is the majority today. For Apis mellifera, we have 2 existing mating places. It's one here, it's on the island Lurö in Lake Vänern. It's one of the biggest lake in Europe. And the other one is here at the Swedish west coast at Väderöarna, also islands out in the ocean in the Skagerrak. And there was another mating place on the Swedish east coast, but this is not actually anymore, or not really working like it was thought from the early beginning, because there was an admixture with yellow bees at least under 2020. This map is not on the actual version. The association is making a new map, but there exist free mating areas farther in the north. Dark brown, they are existing, and light brown, they are planned. But I know there are new gene pools and new plans to change and extend. Something very important in Sweden, we have at the moment no legal protection for Apis mellifera. For those that are not from Sweden, Sweden is a very big country, far up in the north. Our project was mostly focusing on this area here at the west coast, but we also have had one bee yard up in the north and the border to Finland at the polar circle. So 5 apiaries were established in 2019 from the project. And if we're looking much closer, this was here at the Nordens Ark Wildlife Park in Sotenäs at the west coast. The other one was in Uddevalla, where this is a place where I'm living. The third one was up to the Arctic Circle in Juoksengi. This is most far where you can keep bees in Sweden. And the other 2 ones was on the Norwegian side near Oslo in Disneo and Hernes. But I will concentrate my talk on the Swedish apiaries. When we established the apiaries 2019, the colony started with around about the same number of bees, 10,000 individuals, but also 5 to 6 brood frames and 1 honey frame. They was formed in June and August. And we got sister queens for each subspecies and the hybrid, and shared them equally between the apiaries to control the genetic variability. The selection of the queens we have done with help of the beekeepers associations here in Sweden. They are following the Swedish breeding standards like wing indices, morphology of bees, calibrations test on the cake strands, harvest, and so on. But I decided that we want to have for each subspecies 3 different national queen breeders and 1 international queen breeder. So we imported 1 lineage. And those 4 should represent each subspecies and the hybrid. We also installed 1 reserve apiary that was kept under the same conditions like the other one, the experimental apiaries, to make replacement possible, because over such a long time period, there can happen quite a lot. Like I said, 3 queens were from local lineages, and the other one was imported from the UK, the mellifera, from Italy, the ligustica, and from Carnica, Austria. And I also want to have Buckfast from the Buckfast Abbey. But if I was asking for the queen material needed for the research project, I got this answer that I want to read, because we have had even in CICAM quite a lot of discussion about Buckfast. Is it a hybrid or is it a species? But here we have the answer that I want to share with you. Dear Sonja, we don't maintain the Buckfast bee here at Abbey anymore. It isn't a pure subspecies of honeybee race. Rather, it is an inbred hybrid, which is difficult to maintain without encountering inbreed problems. Brother Adam never wrote about these. There are plenty of breeders who do claim to breed a Buckfast strain of bee, but these are a product of technique rather than direct descendants. We are happy with locally adapted mongrels here. We are strong believers that locally adapted bees do better in their own environment and that these attributes don't necessarily travel that well without management adjustments. For example, diseases and viruses have local adaptations and foreign bees don't always cope with a local version of these. Good luck with your project. And if I got this email, I decided, yes, Buckfast is a special case, not a subspecies like the others. And here I get support from Claire. And there I decided not to import something. We took the local Buckfast. The experimental design looks like this. In each apiary, we have had 2 lines with 8 colonies positioned in the same way, the same room between the colonies. In the middle, there was a weather station that was taking the rain, the wind direction, and so on. And after 1 year, we also was changing the positions of the colonies to have no edge effects. And the entrances were colored, but the colored were also evenly shared between the subspecies. So all subspecies got all colors because we don't want to give some in advance or so. And it was mixing after 1 year the position as well. And then each colony stands on a scale system that was giving us the data in a 5 minutes interval. And then I could go into the online platform and looking what's going on there. So it was a really good system, both 5 minutes interval for the weight and for the weather. We also use the scale system to make our notice when we were out in the bee yards and working there. I want to give you an overview how it looks like at the different places. In Uddevalla, it's up here, and here in Nordic at the west coast. You have this Obi fjord on beside the apiary, but otherwise the landscapes are more or less very similar. They have only a small difference in climate because Nordic is a little bit closer to the coast, and there it's a little bit warmer because of the ocean climate. They are similar in flora. They have meadows, deciduous forest, and coniferous forest in the near of the apiaries. And there's no intensive agriculture, no monocultures at either place. It looks a little bit different in the third bee yard, far up in the north at the Swedish polar circle in Juoksengi. There is no sun from the start of December until start of January. Around I wrote midnight, but it must be midsummer. There is sun from mid-May until the end of July. And the cleansing flies are normally from April to the start of May. But in 2020, it could be observed at the 16th of April. And last year, it was already the 24th of March. So maybe shorter winters already start up there. You can see here on this side, this is the Varroa River. It's the border to Finland. And when the bees have done their cleansing flight, they stay for 2 till 3 weeks more in the colony again. When the bee season is starting, the bees need a temperature about 7 or 8 degrees and sun. And this is mostly the case in start and middle of May. Around about. The honey harvest is already done the 5th of August each year. Otherwise, the bees eat too much honey, told me the beekeeper. Here you see how it looks like. Even snow is isolating. The winter starts then in mid of August until mid of April. And they are kept in 2 boxes during winter with 7 till 8 frames, but on the border of the frames, they have polystyrene as an extra isolation, and they get fodder of around 80 till 20 kilogram, but also 5 kilogram of honey. Otherwise, they will not survive the long winter. And you can see here the scale system from a longer period of the experiment. And when we get the scales delivered, it was already October, so we have had no chance to put the colonies on the scales, because there they was already sitting in a cluster and it was too dangerous to make movements. Then you destroy the colony, and that can be the death for the colony already in October. But we put the scales on a kind of a test to see if they survive the winter, but the installation for the scale system was first start in May 2020. And then here you see the honey harvest, and then they got quite a lot of food. Then there's snow and other stuff, which is melting, and then it's the new cycle and so on. Here you see the weather dates from last week. So minus 30 degrees, even during the day, minus 20 degrees. I think now it's also become there a little bit warmer. But last year we make a notation of minus 35 degrees. I was a little bit afraid at the beginning of the experiment that the technique will not make it, but yes, it did. And we are very happy that the system was not collapsing by these temperatures, but it's working well. And the local beekeepers is doing a fantastic job there as well in Juoksengi. But that was the reason why the Juoksengi place was not in the study that we published recently in +1 in October last year, because there we was looking for the first winter. And like I told you just now, the scale system was not installed. But of course, the last winter and the winter is ongoing now is in the data collection. In this paper, we looked at the winter weight loss at the apiaries in Uddevalla and Sotenäs. In total, it was 16 colonies at each place, so 32 colonies both. The total time frame from the experiment was about 20 weeks, 140 days. And we analyzed the time series from the 26th of October until the 15th of March, so one winter only. We analyzed the amount of variance of weight loss and the number of Varroa mites running a factorial ANOVA, where the factors were the subspecies, the colony size class, and the locality. We also developed for this paper different modeling approaches to analyze the weather factors influencing the weight loss. It's known that ambient temperature and biotic factors affecting the metabolic rates of a colony, like in brood or if they're making their cluster for the winter. And several qualitative approaches have been developed, and they're describing quite well the self-organized thermoregulation of individual bees in winter clusters, like they're taking into account the temperature, the individual mortality, the local bee density. But those approaches are not based on empirical data, they are only theoretical models. And ecological processes often show nonlinear relationships. So you're working often with nonlinear mixed effect models, which easily can lead to high complexity and even to interpret which factor is doing the effect of that can become very complicated. So this is why often you found no empirical data. But we make a try, my colleague Niklas Norström and I. We said that each winter cluster was threatened as a superorganism instead of considering each individual bee. And we developed 3 different models: the daily consumption model, analyzing instantaneous weight loss, the total weight model, due to changes of the weight and the nonlinear temperature changes over the period, it is difficult to integrate the temperature in a metabolic function. And therefore, we used a 4-parametric logistic function. The weight assess model, where we modeled the development of the weight in each colony with a constant of daily consumption rate for the superorganism to predict the daily metabolism. Here you only see the total weight model, because I don't want to go too much into the models, but it predicted very well most of the large-scale patterns that we could observe for all beehives. You see, one is a little bit an exception, and you see UD is for Uddevalla apiary and NA for Nordic, and then you see the subspecies behind there. And this was an exceptional one because here the weight was going down very quickly, but this was a queenless colony. I will come back to that later on. Our total weight model delivers more details analysis of the bees' resource consumption strategies. And you can see it here for those that are not familiar with this kind of box plots. It's an interquartile range that you can see in these boxes. The black horizontal bar is a median value. The diamond is a mean value, and these black dots are the other outliers. And now you see here some letters up there, and the A and the V are the lower and the upper horizontal asymptote in this model. And Apis mellifera showed the clearest differences. Its level rate of weight change in the S, you see it's a little bit higher here than for the other three. And if we're looking at the M, even here, it's not overlapping with Buckfast and ligustica, and a little bit overlapping with carnica. We draw the conclusion that this is suggesting a more conservative resource consumption strategy like the other ones. Even if carnica had a late half daytime, so a high M, you can see it here, it has not shown a higher rate of weight changes. The S is lower, like you can see here, like in Apis mellifera. So Apis mellifera's conservative resource consumption strategy could be indicative of an adaptation to a colder climate where it can lower the risk of resource exhaustion. The other models we did not explain here, but we also could find a correlation of light intensity and temperature that influence the weight loss. If we're looking closer to the winter weight loss, the average daily weight loss was really low, like you see here. All colony lost less than 9 kilograms. That is not the normal weight for our climate here in Sweden. The only exception was in the first winter, the queenless colony that lost nearly 12 kilograms. You see it even here, this colony of ligustica, and here the outlier. At Nordic, the weight loss was slightly higher than at Uddevalla, but this factor was not significant. But the effect of size class, so how big the colony was significant. You see it here. They differ quite a lot, a small colony, a medium, and so on. And ligustica and carnica showed higher weight losses in larger colonies than mellifera and Buckfast in the same colony. We transformed this data, of course, to stabilize the variances, and it was an unbalanced design in the different size classes and subspecies. We also did in autumn the Varroa counts, the survey of Varroa destructor infections per colony. In Sweden, we do it once when the colony is without brood. It always depends on the season and the weather, but often it's in November or December. We put then 20 till 35 milliliter oxal acid in a 3.2% solution and drop this between the frames. The amount depends a little bit on the size of each colony. And the Varroa grades were placed at the bottom of each hive for 10 days, and then we are counting them. At the same time, when we open the hive and looking into and making this treatment, we also making pictures and looking how big is the size. And we defined a small colony that covered less than 1/3 of the wax frames. A medium colony covered the half of the wax frames, and a really large colony covered more than a half. And when we was open the colonies, we already saw that the special case of ligustica was queenless, because they are spread over the whole colony. They are not sitting in a cluster, and they're moving quite a lot, so there is no clustering. But anyhow, this colony survived the winter. The Varroa counts was in general very, very low, so below 100 in the mites. But there were 2 exceptions, 2 small colonies of mellifera from a lineage that was coming originally from Umeå. They have above 100 and one has 475, like you see here, the outlier. And one important colony also from the UK has had over 100 Varroa mites, but the sister colony was at Uddevalla with only 62. So we get a significance in the subspecies, but there was no effect in the class of the size class of the colony and the locality. So our conclusion in that paper were that there was no correlation between weight loss and the number of Varroa mites. And we see here something very interesting with these Umeå lineages and with these 2 colonies, because the Umeå area was the last Varroa-free area in Sweden until 2019. In 2019, the Swedish government has changed the law, and there was less controls when you're moving a colony from one place to another place that did not need any controls anymore. And after this law was changed, also the Umeå area got Varroa infections. But this support the theory that uninfected colony first have to learn and adapt to the stress level of mite infestation. And even if I did not show the data here, because we have also made Varroa mite counts last year and 2020, you could see that the Umeå lineages have, over the whole experiment, a high level of Varroa mites in contrast to the other. We found small differences in the winter weight loss between the subspecies. This could show that mellifera have a more conservative resource consumption strategy. But this is only done of one winter. There is more data to analyze, and we will do so. Will be very interesting. And I also have to mention that the winter from 2019 and 2020, when the study was done, was exceptionally warm for Sweden. There was temperatures between -7 degrees only and +13. So that was a very warm winter for Sweden. And the last winter and even this winter is much colder. So it will be very interesting to compare the warmer winter with 2 other more cold winters. Now I will change the topic and go to the RFID technique, the radio frequency identification that we also used in our experiment. You have a kind of tag or responder or chip, and there exists different out there, the active one and the passive one. We used passive one in our experiment that have no power source. And these tags are read by an antenna. There exist different antennas out there, but the antenna is positioned in the entrance of the beehive and is reading the tag. And then it's registrator in a transceiver, and they're sitting a USB key that collecting all the data. Which information is collecting? Yes, the unique ID number, the time step when the bee is passing the antenna, and sometimes other information, but this is depending on the type of the antenna. We use the antenna that is counting the arrival and the departure, and an unknown phase if it's sitting between 2 different antennas. I can show you some pictures that we have taken. Now the antenna looks like. It's looking like here. It's in the entrance. The chips or the tags are very small, so they maybe have 1 to 2 millimeter. And we put them with a drop of glue on the thorax, on the back of the bees, between the wings, so that the wings will not be destroyed or that the tag is wrong situated. So this is a perfect tagged bean, like you can see here. And Niklas here is holding the black box that is collecting all the data. You have some challenges with this kind of technique. First, it is very expensive. There exist different antennas out there, depending on the costs. It's sometimes hard to find the right glue because sometimes these quick glues are smelling, and you have to find the best way to avoid rejection of bean that getting the tag on the back. You have quite a lot of cables in the bee yard, so you have to lift your feet when you're going around. And it's sensitive for extreme weathers, when it's too hot and even when it's too cold. But of course, there are also quite a lot of advantages with this technique. You're collecting a huge amount of activity patterns from your bees. You have daily activities, trip frequency, trip length. You have live characters. You have you can correlate those activity patterns with maybe stressors or weather activities. And I can tell you, it's really fun to tag bees. I like it, or we like it quite a lot, our team. Here we are in action. You can see me, Niklas and Lotte, where we are collecting. We're using this yellow queen cages, and we taking then the bees into our labs that we were building somewhere very close to the bee yard. And then we put them out of these cages, putting a little drop of glue on the back, putting the tag on, and then we have a kind of pen that is scanning the number, and then we can make the notes in which beehive which individuals belongs to. And we were tagging this kind of bee. We call this gråbi in Swedish, and it's very juvenile bees. They are 24 to 48 hours young, so freshly hatched. They're looking different. They are a little bit more fluffy. They have more hairs, and they're moving a little bit different, like really adult worker bees. So it is very easy to see them, and it has another advantage. They cannot sting you either. So it's very easy to handle in that way. And sometimes we really could see how they hatched from the cell, and then we took them. Here you see where we put them back in these cages, back to the right colony, and then they are crawling around and going between the frames, and hopefully they will have a life as an elite bee and collecting quite a lot of data. We tagged 30 juvenile workers bees that were 24 to 48 hours old in each colony. In Uddevalla and Sotenäs, we tagged bees at 3 occasions: in the start of May, when the season is starting, at the start of July, so after midsummer, and the start of September. And this we have done for two seasons, 2020 and 2021. So we tagged nearly 3,000 bees in total at those places. In Juoksengi, we tagged bees around midsummer, only 1 occasion, but this was around about the same week for each year. And there we tagged 60 juvenile workers bees when possible. So around 1,000 tagged bees, even at that locality. Here you see the first results from Juoksengi. This is the year 2020. We tagged the bees here. You found some registrations. This might be rejections of the individuals, but we have not come to the individual analysis of each tag number. But already after, let's see, 1 to 2 weeks of age, they are crawling around the entrance. And the main fly activity we could already see here with good weather conditions after 16 to 29 days of age, so 3 till 4 weeks old. And I told you that the lives or the season at the Juoksengi place is very short. The honey harvest is the 5th of August. And of course, this is a really heavy thing that happens, and you kill quite a lot of bees, but one tagged bee at least was surviving. And this was 49 days after the tagging. I also put some weather data from the scale, so you can see there is rain and bad weather periods when there are not so many fly activities. But of course, we will go deeper into the data and analyze them. The same here, oh sorry, this was too quick. The same we did at Uddevalla. Here you only see the results for 4 entrances only. Even here we was tagging, and then you see the first tagging period was covering quite good 2 months, so around 8 weeks in spring with quite a lot of fly activity. And then the beans was more or less empty, so they died. No more taggings. Tagged bees were registered from the entrances. And we tagged again in the beginning of July. And then there was a second peak of flight activities in the second part of the summer, so the late summer. And then this period was around 6 weeks. And then the third cycle. But here the same with the weather. You see, it's not exactly, but if there's rain, they are not flying. Not so much, at least. Of course, sometimes they are also coming out and doing smaller trips, depending on the wind, especially also. And the third tagging was done in September, and it's shorter because then September can be a really nice period, and there is flowers out there. But then in October, the light intensity is decreasing quickly. And you see there maybe was a sunny October days, but then there nearly no are some registrations. And then we deinstall this technique because it's so sensitive and there's so low activity that it's not worth with this expensive technique to have this left with the snow and bad weather conditions. But we did some test runs already in September 2019 and spring 2020 with 52 tagged bees on 2 colonies. There we was testing a little bit juveniles and adults to get the real handling and so on. And we reinstalled the technique after winter from the 21st of March to the 2nd of May. And this period, we found 4 individuals that survived the winter. We did the same with the tagged bees, of course, 2020, and was reinstalling the technique in 2021, about 120 individuals in 4 colonies. And we reinstalled already in 28th of February because the weather condition make it possible. And we found 1,720 registrations of activity of 40 individuals. So this means that worker bees from autumn in Sweden live up to 6 till 8 months, maybe longer, because we have not checked each individual if we will see the individual number even in from May up to the rest. And this data, hopefully we can extend because we tagged the bees last September and I want to reinstall it in this spring once again. Now I will change the topic for the last time. We also collect pollen. Each colony has at the bottom a pollen catcher, and we activated the pollen catcher for 24 hours with good weather conditions, no rain. Each second week between April and mid-July, we was collecting the pollen, and after each third week after the middle of July to the start of October when the flowering season is finished. Of course, this period was shorter in Juoksengi. There was pollen collection from the end of May to the start of August. We make botanical walks and inventories in the summer 2020 at both places. And the local beekeepers also make notes about the flowering time in each bee yard. And now there are over 1,100 pollen samples to analyze by hand. And this is done by one of, or I would say, Sweden's best pollen analyst. It's Dr. Aslak Dahl at Gothenburg University. She is doing this by classical microscopy. I show you here a picture from Carnica, Buckfast, Ligustica, and mellifera. This was a pollen sample at Nordens Ark in the 25th of August. And she is taking 2 grams of each sample, dissolving this in alcohol, make different centrifugation steps, and make preparations for the microscopy, and then determine and count the pollen so that we know the diversity index. What is the diversity index for ecologists? It depends on 2 parts. You have the species richness, so the number of species in the community, and the other measurement is the relative abundance, the proportion of each species that represents the individuals in the community. Ecologists use many different tools to measure and compare diversity in communities. There exist dominance indices and information statistics indices. And for pollen, you use this Shannon-Venner index. I will not go into all the details, but you have the number of species that you multiply with the natural logarithm of the relative abundance. And the higher the value, the higher is the diversity in that community. But if you're analyzing pollen, you use another index. It's called the Shannon Equitability Index, where you use the Shannon index from here and divided this by the natural logarithm of the number of species. It's also called evenness, and it simply refers to how similar the abundance of different species are in the community. And here it's like it is highest when all species in the sample have the same abundance. So if you have 40 foxes and 1,000 dogs, it's not such equal. So, but if you have 500 foxes and 500 dogs, it has a more evenness. It's maybe easier. And now we got the first preliminary results from Juoksengi. This was taken by Aslak Dahl and her master student, Beatrice Svensson. They analyzed nearly 80 samples collected at 5 different time steps at Juoksengi from all these 4 subspecies, or the hybrid and the 3 subspecies. And you can see around midsummer, all species have the highest diversity. For mellifera, it's very low at the beginning and it's becoming bigger. And if we have passed midsummer, it has the highest diversity, where it's going down for the other one. Buckfast is here the highest. The evenness is the other way around. It goes from 0 to 1. So 0 means it's very different, and 1, it's equal. And here mellifera is here, and then it's going up there. And in this graph, you can see the confidence interval on the y-axis. And it's a high variability here at the beginning for all 3 subspecies, lowest for ligustica, but much lower for mellifera, and it becomes a little bit higher in the end of the summer. If we interpret these results, the pollen diversity varies over the seasons, is highest about midsummer, like I said. And Buckfast as a hybrid showed the biggest variability. So it showed the highest confidence interval in this early summer period. And Apis mellifera was less diverse in spring and summer, but more diverse at the end of the summer. So we can state here that fewer species of pollen dominated the samples, but in a high abundance. And the Aslak found around 30 different species, like Filipendula ulmaria and different Salic species, Rubus idaeus and others. And we stated that it might be a more focused and stable collector of pollen over the short flower period at the Swedish polar circle. So it's a kind of adaptation to the harsh environment up there, because you have not such much flowering like in southern parts of the country. While in Buckfast, It's not kind of adaptation like for the other species or subspecies. It's a hybrid, and it looks like it is collecting whatever it can get. But we will see how this analysis will look like for the second year and even for the bigger bee yards. We also looked into the honey samples, the amount of honey, the water content, and even asked like if done the pollen analysis. And we will look how this looks different between the localities, the subspecies, and the lineages. We're sitting just now with those data and analyzing them. These pictures have nothing to do with the project, but it looks anyhow, interesting and nice to have the honey and see a full honey frame. We also have collaboration with the Plymouth University, because we are looking for the phenotypic data, and Victoria Bushveld, the PhD student from Marine Knight and Plymouth University, is looking at the genotypic informations, and genomic information with phenotypic monitoring will allow identification of the regions of the Apis mellifera genome, and that hopefully is associated with a kind of trait like we observed. And if you have missed Victoria's nice talk, then you should go back and listen to it. And we're really keen to start collaborating now and analyzing both data sets together. I can also mention that our project was high in the media. There's quite a lot of interest out there of the general public in bees in general, but especially for our project, and journalists was very keen to see how you tag a bee and how it works and how it looks like and so on. So there was a huge interest in all media channels. We also make public viewing in the wildlife park, and through the project, we was also able to finance a new observation hive in the park that was already installed very at the beginning of the project, where the people can observe how the bees go in and out, and we also inform in the observation hive about bees and the project. But we have also had an exhibition about bees in general and our project in Nordic and at the University, and of course we were guiding persons around and explaining what we are doing, but unfortunately not in the extent that we would like because of the corona restrictions all over the years. And that is also the reason why the project was extended until June 2022. So now we have more time for the data analysis, for cooperations, and for publishing, of course. And it gives us the opportunity to communicate the results better. We planned now and decided for this day to make our own INTERREG Beestands conference at the end of May to the 2nd of June. And you are very welcome to join us. We will have a webinarium and we'll have parts in English as well about the next results. You can follow us on the project's homepage. We have one at the University of Skövde, and there's another one at Nordic. And finally, I want to thank especially to the enthusiastic beekeepers, Bosse, Anders, and Lotta, for the enormous and fantastic teamwork we have had during the hard work over the last 3 years with such amount of colonies and bees. Also, we have had different students that join us and in different ways. I also want to thank the honeybee queen breeders from the foreign countries that was giving us opportunity to get lineages and fill in all the papers and stuff to get them transported to Sweden. I also want to thank the Swedish beekeeper associations that support us with the bee and queen material and the discussions and further information. I want to name the Nordbiförening, of course, the Svensk Buckfast Avel, the Ligustica Gruppen, and Svenska Carnica Gruppen. And I want to thank my family that hold out with me and my crazy research and one and another bee stink, and that is daily sharing our garden with many, many honeybees. I also want to thank my brilliant colleague, Niklas Norström, for all these stimulating and great discussions about data and data analysis, but also on bee. And of course, the project owners. And here I want to name Mats Niklasson, Nara Nilsson, Ulrika Alroth, Aron Rosmels, Susanne Geisen. In Norway, Louise and Björn and Camilla. At Umeå University, Natushka with her cooperations partners, and in in Gothenburg University, Aslak Dahl with all her students and lab assistants fighting with all the pollen daily. So thank you all. It was a great team, or we are a great team, and now I want to thank you for listening, and I'm very curious to hear your questions. Thank you very much.
Thanks, Sonja. Thanks for a really interesting talk and presenting lots of data from your studies. We've already had some questions that have come in the Q&A section, so I'll just start by progressing through those questions that have already come in. So the first question is from Matisse Heremans, and he says, were the bees treated before you did your Varroa check?
No. We were treating them at that moment where we opened the colonies and making the pictures with this oxalate. That was the first time where they were treated. And then we put the Varroa grade under and counted them after 10 days. And this is normal in Sweden that you only make one treatment.
Okay, thanks. And Matisse has also asked another question. And he's asked, around the polar circle where it's 24 hours daylight, do the bees fly all day round?
If I was at the polar circle, I can say nearly yes, but they also have a short moment where they are not flying in the same intensity, maybe one or two individuals in the entrance or something like that. They have a higher intensity, of course. They have to use the chance that they can go out. But I haven't seen 24-hour cycle in the data where they are flying all the time, but the time frame is longer, of course.
Okay, it's really interesting to see beekeeping so far north.
Yes, it is.
We have another question from Dorian Pritchard that's also about Varroa treatments. And Dorian asks, is it necessary to treat your bees with oxalic acid? What happens if you don't do this? And he notes that he doesn't treat his bees in northern England.
Yes, I know that maybe you can help me, Niklas, but there was one experiment at Gotland, I think, where they haven't treated them. Yes. A study some years ago where Varroa was coming to our country, and they make a test run, and I think some of the colonies, I'm not exactly know how many, they survived. They was not so productive, of course, but it was not the death for those colonies. I have not tried by my own, but I have no idea what's happened, but for sure, from this Gotland experiment, I think they survived. But I'm not sure which subspecies it was or which lineage they use. I think it was not mellifera, but I don't remember what was there used in that experiment.
Okay, thanks. I just noticed that someone has just raised their hand to ask a question, but I think all questioners need to type their questions in the chat box because I don't think you'll have audio at home. So if you type your questions in the chat, I will read them out. There aren't any more questions right now, but obviously it takes some time for people to type in their answers. So while we wait for more questions to come in, I just had a couple of questions related to your total weight model analysis and so on. Do you mind just talking us through the figure again where you had the A, M, S and V? Because I missed some of the details there, and I thought it might be helpful just to have another run through that figure. What were the A, M, S and V parts of the figure indicating?
Let me see which figure you mean.
It was when you were looking at the modeling of weight loss.
Yeah. So M is the date at which the sigmoidal function is halfway. So basically, you had an increase. You had the sigmoidal shape of the weight change.
This one
in that model. Yeah.
Yeah, that's right.
So in this figure, a higher M means that it's a later date at which you're at the initial point of the sigmoidal function.
Okay.
And these are the random effects. So you have first you have a fixed model, and these are the random effects picked out by the different subspecies, organized by the different subspecies.
Okay, so the interpretation is it takes longer for mellifera to lose weight than the other subspecies, and overall they lose less weight than the other subspecies.
Yeah, the whole sigmoidal shape is moved a little bit later, so the change is later.
Okay, and so my follow-up question to that, while we wait for more questions to come into the chat, was obviously you've observed this effect, but are you able to explain it mechanistically of what the difference is between the subspecies that explains these different patterns in those sigmoidal curves?
Well, as we could see in the results, for example, that M was higher for mellifera, and also that S, the next one, which is the slope of the sigmoidal function, it was higher here, which in this model means that it was actually slower, the slower rate of weight change. It depends on how the A and the B values are arranged. You can get it to be the opposite as well.
Sorry, in terms of the physiology of the bees. Yeah.
So we haven't linked thoroughly to that, but it could be like more hairs, for example, or something else that can keep insulation so that they do not need to spend all their resources to maintain heat, for example. That would result in a slower decrease, for example.
Okay, thank you.
But we didn't put too much effort into that.
Yeah. We've had some more questions come in now, so I'll turn back to the audience. So the next question comes from Mary Knights. She says, thanks for a great talk, Sonja. It's an amazing project and it's so useful to have these kinds of data being collected. Did you get any impression of whether there were subspecies differences in colony buildup in spring or in temperament? And that question comes from Marianne Victoria.
Yeah, we have them on the scale system, but not analyzed the data yet, how quickly they increased in spring. But mellifera is a little bit slower in the spring to starting, in contrast to carnica, for example. But we will go further into the data and see if the data will show us and be the same like our local observation when we were working in the bee yard. But the other way around, if you're looking over the whole summer, Apis mellifera also is collecting quite amount of honey in the late summer because they go to the common heather, this typical Scandinavian flower. And there they can be very productive. And I can tell you in the season 2020, they was collecting up to 45 kilograms per 24 hours, and none of the other species was able to make that.
Oh, wow.
It was very impressive. So they are much lower in the spring or at the beginning of the season, but they can be so productive at the end of the season. And I was very happy to see also this pollen analysis now from Aslak, because it show a little bit this also at the end of the summer, that they are more diverse there, where the others already go down and seem not to collect more, because it's maybe really cool and an adaptation to our environment.
Yeah, that was really interesting. That diversity seems to be driven by more evenness rather than foraging on a greater number of species.
Yes.
Yeah.
But the diversity anyhow in the pollen was higher. It's the second part of the summer. So at the end of the summer, even the diversity was higher there, where the other species already were going down. And this should also explain when they really collect such amount of common heather, that they are out there and they collect in the last they can get before the long winter is coming. It looks like that. In temperament, we have made notes, but we was not so focused on aggressivity or something like that, because we think this is a kind of very not so objective, because if I think it's aggressive, maybe you don't think it's aggressive. So we was not so into that part, more on ecological and biological data.
Okay, thank you. We've got a few more questions to run through. Another one from Matisse Heremans. He asks, how big were the winter clusters between the subspecies?
The winter cluster in size, or how does it? I don't get it. What you want to know? We noticed the size of the cluster where we could see it when we open the hive and make pictures and say, okay, it's a big one, a small one, and so on. I said at the early beginning that we also make thermography, photography during the winter cluster. We are just now working on that data, where we really see in the heat pictures how big the cluster is, but it's not so easy to analyze. So we are still working on that. It's quite a lot of pictures once per month per colony, and you see that it's shrinking and that they are moving in their position, but we cannot say yet if there are different strategies, how they go over the frames also.
Okay, yeah, I think then Matisse has confirmed that that's what he wanted to know. So I guess wait for the extra data to hear more about that at another conference in the future as well. The next question is from Vincent Doir, and he says, hello, Sonja and Nicholas. Thanks for this very interesting talk and for your presentation of Sweden honeybee reality. The question is, how are beekeepers dealing with admixture? Sorry, I don't quite follow the second part of the question. I think, yeah, sorry, how are beekeepers dealing with admixture, and have you conducted an overall study of admixture in these bees?
Yeah, I think all these associations are looking with the wing morphology, and they try to keeping their mating places as free as possible. Therefore, we're using these islands out there at the west coast, and even other associations are using that. We have these wonderful big islands in the oceans, but anyhow near the coast. But it's quite a lot of work to move your colonies and queens there and getting them mating in those places. But it's a very good possibility to put them on islands and do it there. But if you have not such strict possibilities, then of course you can go for admixtures. There are a little bit, please correct me, Niklas, in the south it's more, some subspecies are in some regions more dominating than in others, and people try to get free mating areas. But I said at the beginning, we have no local protection for none of the species, or so like in maybe other countries. So, and that make it, of course, more difficult for the beekeepers to keep their species clean. Well, Niklas, you have something to add?
No, I agree. I think since you all also have the different sections in the north of Sweden, so I would say that there are different parts of Sweden where they try to get breeding areas, pure breeding areas.
Okay, yeah, thank you. Let's move on to the next question. Oh, well, there's a follow-up question also from Vincent. It says, concerning the adaptation of the honeybee, how amazing to see such low differences between pure breed from Italy and the UK. And did you monitor locally adapted colonies? I think that question goes back to your experimental design at the start. You had your international subspecies and the national colonies from the national breeders as well, didn't you? That's correct.
Yes, that's correct. We have not gone into the data and see closer if these imported subspecies will be or have another strategy like the others, because we were saying we wait until we have the whole data set, because then we have more replicates from year to year. But like I said, in this Varroa case, we could see a little bit that some lineages were different if we moved them, but not from the imported one. And even in that paper, we got not into that yet, but we will do that, of course, and analyze the data, because that was the reason why we imported one lineage for each subspecies, one lineage. So that will be another interesting data set that we hope we can show you at our conference.
Mari's just made a note in response to the question that she asked, saying, yeah, it was really interesting to see the difference coming out in the data at the end of this season for the, that's in relation to the species richness, the evenness, diversity analysis of the pollen. There's another question from Balsa Fried. Let me just read the question. It's to do with winter feeding. At which time, at the beginning or end, do you give the honey? Or are you mixing the honey with sugar syrup?
Okay, if you do the honey harvest, like in Juoksengi, the 5th of August, or we did it at the end of August or the beginning of September, you're taking the honey and you keep them, some frames, around 4 to 5 kilo, depending a little bit, or 1 or 2 frame, depending how good it's filled. And then you directly give them the sugar water. And this is a very short period because it has to go quickly. And then they fill it in vessels. So a maximum of 1 week or 10 days in the 2 lower bee yards. And in the upper bee yard, I think it's also a very short time frame, because you cannot wait quite a long period, because the weather is really the killing off. So we do it in 1 week. So each second day you are filling the feeder on the top of the colony, and then it's quite a lot of activity and hopefully good weather that they can take everything and you can fill up. But it goes very quickly, and then all the food reserves are filled, and then the winter can come. It's a little bit like this.
Okay, yeah, we've not got any more questions right now, but again, while we wait for people to type in questions, because sometimes obviously can take a while people type, I had a few more questions myself. So you mentioned at the start there's no legal protection for AMM in Sweden at the moment, and obviously protecting AMM is quite important to the goals of the organization. So I just wondered on that note if you could tell us what the prospects are for the future. Is there increasing political interest in local native subspecies in Sweden? Are you hopeful for the future about protection?
From my personal perspective, I'm very hopeful. I hope that we can be a part in the process as well. And you can see from teaching newcomers and beekeepers that there is a huge interest for Apis mellifera, and the people are curious, they want to know. And I think there is another knowledge about local adaptation and biodiversity, and the people want and help. And it has to be done on a good and correct way, of course, but we also would like to have maybe the Swedish Agriculture Board on the same table and discuss this and why and how. But by myself, I found it very interesting to hear from other European countries that they have become longer. And Sweden is a big country, so I think we have the power and the possibilities that we can get some places or areas at least, hopefully protected somewhere in the future. And I mean, it's not a problem for Apis mellifera. Other associations have the same problem with admixture and inbreeding problems and so on. So at the end, I think we're sitting all in the same sinking boat if we not get something done in that area. That is my opinion.
And well, maybe just one final question from me to draw the question into a closer, just in the interest of time. But my question may be slightly tangential, but in your biography, you said you were interested also in climate change and species distribution modeling, and obviously you presented data this evening on honeybees really far north. And I noticed there's one day, I think, where it's 30 degrees and there was almost double the amount of visits on the next highest day of 2000. Can you tell us more about what the likely climate change scenarios are for Sweden in the future, and how that likely affects the prospects of beekeeping throughout the whole of Sweden?
When I was running this climate models, it was for marine species, but I think, but it doesn't matter. I mean, the conditions are the same, that the species will move farther north, and it will change, of course, the water getting warmer. We can see and observe that in the ocean, and then it starts at the terrestrial environment as well. We have had quite a lot of hot summers. The 2018, for example, was very hot in Sweden and very dry. And if you have a very dry summer, you know, as beekeeper, this is not good because bees need also water. And the flowering period was very short. Each flowering period was extremely short because of no water. A country like Sweden that normally has quite a lot of water was not expecting such a scenario, but it happens. And I think that will be, of course, the future, and it will be challenging. Of course, and I mean, even this beekeeper, Busse, that joined our project, he is doing beekeeping at the Polar Circle for 40 years, and he lost quite a lot of colonies last winter, and he did not know why, but maybe the reason, because the wind was coming from another direction than 20 years ago, that can have really extremely effects in not only for marine species or others, even for the beekeepers. And we do not know. The scenarios always are so good as you put the data and models on, and you have quite a huge of variability, but you can already now observe changes. And I think that quite a lot of beekeepers will agree with me that there was some difficulties the last seasons, and I mean 5 years or so around. So what are you thinking, Niklas?
Yeah, no, we can maybe link this also to, there are a lot of differences, but I was also thinking about linking this to the weight change paper, because we saw when we looked at the environmental parameters, it was temperature and light intensity that was mostly driving the weight change from the statistical analysis. And if they were in interaction, they had the best explanation, or the strongest explanation. And if they are if you have them in interaction, and if you have temperature changing, but not light, then you switch that into interaction between those two. So that could be one mechanism that could impede maybe the adaptation towards another new climate.
Okay, that's really interesting. And if I remember correctly, I think 2018 was the last time we met for an in-person conference with SICAM, and it was in Finland. And I remember some really nice, warm evenings sitting outside with a beer talking about honeybee conservation. But I remember being surprised how warm it was up there in Finland in that summer. I think it was, I remember the real heat wave all across Europe. We have one final question to end things with. It's from Lars Andreas Kirkerud. He says, thank you for your interesting work on differences among species. Did the study of flight activity reveal differences among subspecies?
Yes, we have not analyzed the data yet because it's enormous data. But like I said, you're welcome to join us after the conference, and hopefully we can present there the data. So you're welcome. And maybe there's a possibility that we can spread an invitation with the SICAM Foundation, and you are all very welcome. And then hopefully can hear more analyzed results of our data. So, but for sure, we will keep a while to analyze all the data because they are a big amount.
It's really impressive, the amount of data that you've collected. And that was a really fascinating talk. So thanks for your time this evening presenting to us. And thanks for the answers to all the questions. On behalf of the audience, I would just like to thank you. And we'll leave things there. So thanks again.
Yes. Thank you very much.
Thank you very much.
Yeah, and thank you very much. And
see you.
My technical problems are solved, so I can now normally talk again. I had 2 windows open, so I heard myself 3 times, I think. So
yes,
I excuse for that, but now everything is okay. So I want to also say goodbye, and maybe till next week.
Yes, of course, we will join.
Yes.
Thank you very much. Have a good night, evening.
Bye-bye.
Bye-bye.